Hey guys, I just wanted to touch base with you and see how you're doing with chapter 17. In order to keep these videos a little bit shorter, I got a little carried away last time and had to rush through. I wanted to do a couple short, quick videos. So this is going to be kind of a general overview of histones that I kind of breezed over yesterday. And I want to introduce you to transcription and translation. I'll post a few more videos on this tomorrow and this weekend. But for now, just go ahead and kind of get yourself familiar with a lot of the key words and a lot of the vocab. These last two chapters, 16 and 17, have been very intensive, right? We're talking about replicating the DNA so that we can make more of it and go through processes like mitosis and meiosis. And now we're going to talk about pretty much making the foundation of life, you guys. Proteins, we know in the cell, do what? Awkward pause for everything. Proteins are the foundation of everything, and the way they fold and bend is really important. Proteins like to bind to stuff, and when they do bind to stuff, they change shape. They can be denatured by things like pH and temperature, so they're really critical to how the cell functions. So this chapter is transcription, how we're going to write the code for making proteins, DNA to protein, because that's the point, right? DNA is the recipe for life. So what are we making? What's the end goal of our recipe? We're cooking up some proteins. So DNA to protein, transcribe it, and then translate it. I want you guys to think back to all of the analogies I've used about cookbooks, right? If DNA is the cookbook and we've locked it away in the nucleus, what we have to do is take grandma's version and put it into a shorthand, right? You don't want to carry around grandma's big book of recipes. You need to put it into a condensed file, something quick that you could access very easily. So the whole point of transcription and translation is going to be to get that long, complicated DNA code into something a little bit more manageable. Why do we need to do this? Well, number one, we've got to get it out of the nucleus. In eukaryotic organisms, we keep that DNA inside of our nucleus. This is a little bit different for bacteria. Bacteria can transcribe and translate all at the same time because they really don't have that nuclear envelope to hold things in. But for us, we've got that lockdown safe, right? That nucleus. The nuclear envelope does have some pores, but the DNA is too bulky. You guys just learned in chapter 16, we've got double-stranded helical shape. It's not going to go through those nuclear pores very easily. So what we need to do is get it into a form that can sneak through those nuclear pores out into the cytoplasm of the cell. That's where transcription comes in. We're going to take that ladder structure, that double helix DNA, and instead of unzipping and replicating itself, we're actually going to start on these TATA boxes. They're going to be these start codons. You're going to hear words like codons. They're chunks of 3, right? We're going to move in groups of 3 from 5' to 3' because we learned last chapter that it's easier to move down our steps. And we're going to kind of start at these start boxes, wherever they are on a gene. That's going to be the start of the protein we want to code for. We're going to come along, we're going to kind of open up that double helix, we're going to unzip it, but we're not starting from end to end. It could start anywhere on that one strand of DNA. We're going to unzip it, and in that one little bubble, that area that we've unzipped it, we're going to make an mRNA. M for messenger. That's going to be the condensed DNA version that we're going to transport out of the cell through the nuclear envelope. So we're going to make this mRNA. So apples grow on trees, cars go in garages, except mRNA is a little different, you guys. It doesn't have that T, that thymine. We're going to replace that with uracil. So apples end up becoming applesauce. That's how I remember it. And when you're making an RNA, instead of a T, you end up with a U. Apples end up as applesauce. So in this case, everything will still be the same. A's will go with U's, C's will go with G's, and vice versa. And we're going to make a single-stranded mRNA, messenger. It's going to be the messenger carrying this condensed version of DNA, a condensed code for this one particular protein out of the cell nuclear envelope and into the cytoplasm. Once it gets out of the cytoplasm, now we're going to translate. It's like you taking that big, awesome grandma's recipe cookbook, taking it to Nabisco and saying, okay, let's make a cookie for the masses. Once you get out into the cytoplasm, here your ribosomes are going to come together. They're going to make like a hamburger, a large subunit and a small subunit. They're going to jump on top of this mRNA, and then we have something called tRNA, transfer RNA. They're going to bring pieces of protein codes for us down these amino acids. They're going to bring these amino acids, T for transfer, and whatever codon that ribosome is reading, it's going to add an amino acid for that particular segment. Remember, you guys, however we put together these proteins determines what they're going to do, either enzymatic, or if they're going to act as units of the cytoplasm, they're going to end up on an endoplasmic reticulum, how we code for those things. So for right now, we're just keeping it simple. I'll go into a more detailed video later, but we made mRNA, a condensed version of the DNA. We transported it out of the nucleus into the cytoplasm, where now we're going to go through translation. We're going to take that mRNA, we're going to read it, and a transfer RNA is going to bring us the appropriate amino acid for every little segment that it needs to code for. There's an awesome chart that we're going to learn how to use together when we get back from spring break, where you can actually read. It's the same amino acids. And you guys, these are the same amino acids for every living cell. It's just how we put them together. So bacterium, fungi, plant, animal, you name it, we all use that the amino acids listed on that chart. So we'll go into that a little bit more in detail later. But you guys, transcription takes place inside the nucleus. We get it out through messenger RNA. Translation takes place in the cytoplasm using our ribosomes, large and small subunits, and tRNA, transfer RNA, that's going to act like a caddy at the golf course. He's going to bring us some amino acids that code specifically for each group of 3 that we're going to work with. 3 nucleotides will code for 1 amino acid. We'll practice this a lot more later. Before I go, let's talk a little bit more about histones, you guys. We breezed through it on the last one, but you guys, histones are basically a majoral structural protein, okay? The DNA, once we go through replication from chapter 16, we end up with so much DNA inside of that nucleus that it's going to take over, right? We've got all this DNA, it's packed in a nucleus. We need to organize it before any mass events like mitosis and meiosis can take place. Otherwise, things may get complicated and twisted, and you saw those double structure, the double helix. We need to keep it under control. So the DNA will actually wrap around those histones. And the histones, you guys, are just more proteins. Now, there's an order that they go in, right? They wrap twice, and then they keep coiling until they get further and further coiled. You guys don't need to know that it becomes a nucleosome and then a solenoid and then a scaffold. Nothing like that. Just know that histones are really important for condensing that DNA and making it into chromatin, that tightly wound structure that's about to go through something like mitosis. But we have 2 different types of chromatin, you guys, even on the same chromosome. We can have heterochromatin, which is generally not very active. It's very, very compact, very tightly compact on that chromosome. And then euchromatin is a little bit less compact. It's not as dense, and it's very active. Why would you have 2 types of chromatin? Again, it's just how tight that histone winds that DNA. The reason is you don't want it to read all of the time. You want to start that replication where it's going to be less active. So typically towards the end, toward the telomeres on your chromosome, you'll actually have it be a little bit more heterochromatin based, which is going to be tightly active. Near your centromere, near the ends, you don't want a lot of replication taking place. But on the middle of those chromosomes is where you'll have your euchromatin, where we can have a little bit lessly dense and more activity for replication. So that's a little bit more detail into our chromatin and what that does. It's going to be wound up by histones. Histones are just proteins that are going to bind together, and octameric systems in groups of 8, they're going to be beads on a chain of DNA. But it's really important, you guys, because once we go through replication, we need to get that DNA under control. We've made so much of it that we need to make sure we can get it organized again. So when we do things like transcription and translation, the DNA is very organized in fashion. I hope that helps. I'll post another video that's a little more detailed on transcription and translation and RNA polymerase and all that good stuff a little bit later. Take it in chunks and do lots of vocab study. If you guys have any questions, just shoot me an email. Bye.